are going to cause further development, and in particular, the development of a limb. Now, in different scenarios, we have to look at how these processes are actually regulated larger scale, not just in a single individual. So we can look at something known as a homeobox gene. A homeobox gene is a very, very small 180 base pair. That's it. That means you have A, C, T, G, but only 180 of them, okay? And in this sequence, what we look for is the regulation of all other developmental properties. So essentially, when we look at a homeobox gene or a homeobox sequence, we're actually looking essentially at the construction foreman. Or forewoman if you prefer. Okay? What we're looking at is the person in charge, right? When we talk about somebody like a construction foreman or forewoman, we're looking at someone in charge. They don't necessarily get their hands dirty in the process. No offense, but they don't necessarily get their hands dirty in the process. But what they do is make sure that everybody else does their job right. That's the job of a homeobox sequence. It's to make sure that every gene, every morphogen that is regulated and working on this developmental process does its job when it's supposed to and as well as it is supposed to, okay? The interesting part of homeobox genes is that I have here that they're highly conserved. Now, we haven't talked a lot about genetics yet, so knowing that something is highly conserved probably doesn't tell you much. But when I say highly conserved, I mean that I can look at the exact same 180 base pairs in you. I can look at them in a fruit fly. I can look at them in an elephant. I can look at them in a guinea pig. I don't care. Okay? Any animal. But it goes beyond that, we're finding. These homeobox genes are actually highly conserved across not only animals, but into other organisms as well. So when we look at mutations to this, our concern is that we will get improper development. And scientists have worked with this gene enough now and this sequence that if they change anything in this sequence, if they change the way in which this sequence behaves, so it's coding, so the A, C, T, and G changes just ever so slightly, one base pair change can completely change the outcome. From the standpoint that in our fruit fly here, we can get legs where their eyes should be. And we'll look at more information on this later on, but it literally is, you can get different limb structures, you can get different development, you can get different orders of development, you can put torsos on upside down. It's really kind of science fiction, mad science kind of things going on. But the whole idea is that it helps us understand how development occurs. When we change something and we see the result of that, we now know what actually is making that happen. So I mentioned to you that this homeobox gene is actually highly conserved across different species. We've now found them in mice and flies, okay, split about 550 million years ago. We'll look at that in the animal kingdom. So the difference between our mammals and other animals. But they've also now found the same homeobox genes in fungus and plants. Fungus and plant split from animals 1.2 billion years ago. So when we're talking about a 180 base pair sequence that is the same in a fungus and in you, that's pretty amazing. And before you say you don't look anything like a fungus, I disagree. You have a dorsal, you have a ventral, you have an anterior, you have a posterior. Homeobox genes set up a three-dimensional body plan. They don't care what your body looks like. They just need to set up a plan. So in this, we can look at all kinds of changes in different types of genes. I give you some examples here. I'm not too concerned about you remembering Hox and Hom. Some of you may be interested in that. What I want you to remember is that Homeobox genes are highly conserved. And I can take a Homeobox gene out of you and I can put it into a fruit fly and it's going to do the same exact thing. So we start to look at why then do some insects have 6 legs, other arthropods and so on. Why do you have 2 legs? Okay. Why do spiders have 8? Right? Why do giraffes have 4? Why does this occur? So it all has to do with that Homeobox gene, and essentially it's going to act like a light switch. If I want 2 legs, I turn it on once. If I want 4 legs, I turn it on twice. If I want 6 legs, I turn it on 3 times, and so on. Okay? So in this process, what we've got to think about is this is an overarching regulating mechanism. Okay? It's not going to make your toes pretty, all right? It's not going to make your fingers longer or shorter. It will, however, set up this body plan and give you the essential basis for making the rest of you. Okay? So I show you here just briefly in our fruit fly and our mouse. If you look at the color banding, that's what we're most interested in. Now, yes, we've got 4 mouse chromosomes here, 1 fly chromosome here. They're going to have the same information. Flies have fewer chromosomes than mice. So the information is distributed differently on the chromosomes, but the information is the same. When I have yellow bands here, okay, my yellow band associated with my fly chromosome, my yellow bands, or orange bands, pardon, associated with the mouse chromosomes, okay, we get orange here, we get orange here, we get orange here, we get orange here. Well, what does that mean? When I actually come down and look in my adult organisms, I got orange here and I got orange here. What did I make? Probably made ribs in my mouse. I made an exoskeleton in my fly. But I made a dorsal side, dorsal on both sides. I gave it particular definition as far as being part of the thorax and upper abdomen. These are important. This is basic developmental structure. It's a 3-dimensional plan. That's the important thing with morphogens. We've got to elicit change with the morphogens, but we also have to think about the fact that this is not going to give us particular specific details. It's going to give us big picture. Okay? We'll get into more of the specific details with genetics in the next couple of lectures.